How's it going, everyone? It's Chow here, and today we are going to discuss a little bit about gene regulation, so let's get started. Okay, so gene expression, very important topic, I think, but when it comes to gene expression, I think it depends on, how much information you already know, because if you have a good understanding of transcription and translation, you probably already have a little bit of idea when it comes to how you can control the amount of product produced. But when you look at the whole concept of regulating gene expression, it's basically looking at how a cell is able to control the production of, for instance, various proteins. So the cell can do a multitude of things, including downregulating transcription, hydrolyzing the mRNA before transcription, preventing the mRNA translation at the ribosome, and then other modifications afterwards. So what's really key here is that the earlier the cell can stop synthesis, the less energy is going to be wasted, which is very critical, obviously, for cellular function. When you look at the concept, of course, of regulating cell and gene expression, there's really only two types that is going to be presented in sort of an introductory classroom, the first being negative regulation and the second being positive regulation. So negative regulation is just simply a gene that is always on unless a repressor binds to it and basically stops transcription from occurring. On the opposite hand, you have more positive regulation, which means that a gene is going to be off unless an activator protein of some sort binds to it and then basically allows that transcription to actually occur. So these are the two main types of regulation that you're most likely going to see in cellular mechanisms of controlling gene expression. Now, the proteins, of course, that are being transcribed in the process are going to also be different. So we generally categorize them into either inducible proteins or constitutive proteins. And inducible proteins are ones that are basically produced only when needed, whereas constitutive proteins are basically produced constantly. So if we're looking at gene expression and gene regulation, one great example that we can use is with the E. coli model system, especially in regards to lactose breakdown. So again, there are basically two main groups of proteins, but what we're really looking at in terms of the E. coli model system is the operon. So an operon is just a gene cluster with a single promoter, and this operon consists of basically a promoter, some structural genes, as well as an operator region. And what's really critical about these operons is that there are basically three ways in sort of a basic bacterial level that can be used to kind of control the amount of gene product produced. The first one is with an inducible operon, also known in our case as, for instance, the lac operon. Another type is the repressible operon, and in our discussion it's going to be the tryptophan operon. And then finally, we have sort of an operon that's going to be operated and regulated by an activator protein, and this is the example of catabolite repression in the lac operon. So kind of three general ways that you're most likely going to see in an introductory classroom in regards to operators, operons, and controlling gene expression in, for instance, E. coli. So that's really nice and all, but when we're looking at the operons themselves, there's going to be variation. So we talked about just recently how some proteins are going to be inducible and some are going to be constitutive. So the first operon that we're really talking about here is the lac operon. This is a summary of the lac operon. You can definitely read it, but basically the lac operon is really fascinating because in E. coli, when there is lactose absent, so there's no lactose around, basically a repressor molecule is going to recognize the operator, bind to it, and prevent transcription of key enzymes. On the opposite hand, if there is going to be lactose present, you're going to actually induce the production of the enzymes necessary to break down lactose. So that, I think, is pretty straightforward. Again, here is basically a summary of it, and you can most likely just read through it and understand it as a whole. So another type of operon that you're most likely going to see in E. coli is the TRP operon or tryptophan operon. This is a repressible operon, so it's going to be on as the default system, and it's going to stay on until there's going to be some kind of a thing that prevents it. And this thing that prevents it from being continued to be on and producing more and more of the product is going to be tryptophan. So ideally what happens is when tryptophan is going to be absent, you're going to basically not have an active repressor. So the repressor is inactive, which means that transcription can occur normally. But when tryptophan is present, the tryptophan actually acts as a co-repressor and binds to the repressor molecule. And this co-binding of the co-repressor and the actual DNA itself is going to ultimately prevent the transcription of those genes. So it's kind of, you have some kind of an extra amino acid that acts as a co-repressor to prevent the cell from making more of that amino acid. Whereas back in the lac operon, it's basically you have to make a protein to break down something that's present in the surroundings, in that case lactose. So this is actually a summary of what happens with the tryptophan operon, and it might be good to just take a look and just pause it and see if you have a good understanding of this specific thing that we're talking about. Okay, so the last thing that we're really going to be talking about in regards to gene regulation in E. coli is a catabolite repression system. So in the lac operon, again, there's actually a catabolite repression system. And this is actually really fascinating because it's regulated by an activator protein. So that protein is going to be regulated in itself by other external factors, like, for instance, glucose presence. So glucose in the cell is going to be the preferred energy source. If you have both glucose and lactose present in the cell will preferentially produce the enzymes to basically break down glucose. The lac operon, therefore, is not transcribed efficiently because the cell wants to make enzymes that break down glucose and not lactose. So in order for efficient transcription to occur, it requires the binding of an activator protein to that specific promoter region. So this is a case of positive control where you have an activator protein that's going to come in and increase transcription under the right certain conditions. So if you have, for instance, a low glucose level, so this is one of those rare cases potentially where you have low glucose present in the surroundings but high lactose presence. If glucose levels are low, there's going to be a signaling pathway that occurs that leads to an increase in levels of cAMP or cyclic AMP. That cyclic AMP is ultimately going to bind to a cAMP receptor protein or CRP, and the conformational change in the CRP basically allows it to bind to a lac promoter. The CRP here in this case acts as an activator of transcription, and its binding actually results in sort of more efficient binding of the RNA polymerase and therefore increased transcription. So again, this is a situation where we have low glucose levels and high lactose presence. So in a low glucose and high lactose level presence, this is what's going to happen. So you have this CRP that's acting as kind of an activator of transcription. On the other hand, however, if you have, under different circumstances, you have high glucose levels, but lactose is also present, maybe in same or smaller amounts. Again, remember, your cell is going to be more trying to use glucose. So you have E. coli that really wants to use glucose rather than lactose. So even in the presence of glucose and lactose, especially in the presence of high glucose and maybe smaller amounts of lactose, the CRP does not bind to the lac operon promoter, and therefore the efficiency of transcription of those lactase enzymes are going to be lower. This is known as catabolite repression, which is basically a system of gene regulation in which the presence of a preferred energy source kind of represses other catabolic pathways. So again, this is a situation with high glucose and lower lactose, whereas in this instance, it's going to be lower glucose and high lactose. So depending on the concentrations of external nutrients, for instance, like glucose or lactose, the cell might be using different ways and different methods to kind of allow itself to gain energy. So that's basically it when it comes to operons and regulation. Of course, when it comes to regulation, things get a little bit more complicated in eukaryotic gene transcription, because obviously in multicellular organisms there's going to be a greater complexity, and so there's going to be certain protein controls that need to be regulated, sometimes upwards or downwards. So the expression of eukaryotic genes have to often be regulated fairly precisely and potentially at different points. So that's also very important. Of course, proteins can help kind of regulate the process as well, because if you think back to eukaryotic transcription, remember this thing called the Tata box. So in eukaryotes, there's this little section over here called the Tata box, and that's kind of where the DNA begins to open in terms of transcription. Promoters also include regulatory sequences recognized by transcription factors, and of course you ultimately have this unique combination, this complex that forms between the RNA polymerase as well as your different transcription factors and your general transcription factors. And it's this binding that allows for kind of this initiation complex. So here you have transcription factors that can help regulate the transcription of whatever gene we're talking about. Now, just a quick recall, in prokaryotes we have a sigma factor and not sort of this unique system with the RNA polymerase and all these transcription factors. So it's a little bit more simple in prokaryotes for obvious reasons. Now, with this kind of concept of regulation, you obviously can also use other sequences, including DNA, to actually help regulate or, upregulate or downregulate transcription. So enhancers and silencers are usually what we like to term these kind of regulatory DNA sequences. And an enhancer is basically a sequence of DNA that binds to the transcription factors that allows an upregulated activity to either increase or maybe even just activate transcription. Whereas on the opposite hand, we have silencers, and they kind of operate in a similar way to enhancers, but by binding to transcription factors, they kind of repress transcription levels. So these are basically sequences of DNA through mediation by transcription factors, for instance, that can help kind of regulate transcription levels and ultimately gene expression. Of course, there are often other things that are involved in the complex, and it can get really complicated, but oftentimes the combination of factors, present in whatever system we're talking about can help determine rates of transcription, and then although the genes are often present in all cells, so the same genes are present in all cells, the fate of the cell is going to be determined by which of the genes are being expressed. So if you look at genes from different tissues, you're often going to find that they're almost exactly identical. But why are the tissues different? Why is the expression different? That's potentially due to regulation. So which genes are actually being expressed at which location, that can be very critical. So when we look at gene expression itself, a lot of genes can actually coordinate expression of different sets of genes. And that's very critical because most genes have their own promoters in eukaryotic cells, and they might be far apart in the genome. But if the genes have common regulatory sequences, they can be regulated by similar or if not the same transcription factors as shown over here. So when it comes to eukaryotes, you can also not just have specific genes being, regulated by certain transcription factors, but you can actually have certain sets of genes that are going to be regulated either upwards or downwards by transcription factors as well. And this is going to be very critical for gene expression and allowing certain cellular fates. And finally, just before we go, another thing that we can discuss when it comes to gene regulation is basically how they can be controlled. And these, of course, include various opportunities such as alternative splicing. So that's going to be discussed in another video, but alternative splicing of pre-mRNA, kicking out those introns, can ultimately change basically the gene expression. In addition, after transcription and you make your mRNA, you can have translation repressors that can also kind of affect basically how much that gene is going to ultimately be expressed. perhaps what's even more fascinating is you can actually allow for regulation even after translation. So you have translation, you make your protein or your polypeptide sequence, you can actually make even more modifications. And three general ones include proteolysis, which is just cutting of the protein, for instance, removing a signal sequence. You have glycosylation reactions, which are just basically adding sugars to proteins to make a glycoprotein type. And these glycoproteins are obviously very important in the surfaces of cells. And then finally, you can have phosphorylation events, which are also very key in signaling pathways. But basically, by adding certain phosphate groups onto a protein via these things called protein kinases, they can actually change protein conformation and ultimately affect whether the protein is going to be active or inactive. So post-translational modification of polypeptides can also affect gene expression. And then also, just lastly, before we actually end up going, we also have these things called RNAs that can actually help regulate gene expression as well. So lots of things can help regulate gene expression. And RNAs, including microRNAs and small interfering RNAs, can also arise that can help affect the regulation of certain genes. And here's just basically a short explanation of what's going on within these sequences. So bottom line is that when it comes to regulation of genes, you can transcribe the gene and you can make that gene, but depending on the regulation sequences and certain aspects, for instance with operons, for instance with transcription factors, and then even afterwards with post-translational modifications, pre-translational modifications, splicing, etc., all these different aspects can contribute to the amount of a gene produced, whether that's a lot or whether that's very little. And then depending on the needs of the cell, you might be able to shift some of these processes and basically upregulate or downregulate the amount of gene produced. And finally, last but not least, also perhaps just as important, is the environmental factors and the presence of certain nutrients can most certainly affect the amount of upregulated or downregulated activity within the cell and ultimately contribute to variations in translation and transcription and gene regulation as a whole. So there you go. That's my ramble. I hope you all found it useful. Best of luck studying, and as always, I will see you in the next video.